identifying some of these white blood cells, looking at what they do in terms of the kinds of infections that they fight. So, for example, okay, today we looked at just general white blood cell counts, right? Like, was it high, low? So just like with the red blood cell count, you know, the red blood cell numbers, we were looking at that as well. But we also did hematocrit and hemoglobin for red blood cells, you know, looking at our blood. But white blood cell counts, they do tell us generally if we're high, low, or normal. However, to do a differential white blood cell count is going to give us a lot more information, because if we know, for example, a specific white blood cell number is up, that can indicate what kind of infection we have. So that's what we're going to be talking about now. We're going to talk first a little bit about the characteristics of the white blood cells, and then we're going to refer also to the handout as we talk about things, so that you get an idea. And then in class next time, we're going to get our scopes out, take a look at our white blood, or I should say our blood smear that we have in our slide boxes, and we're going to count up the different white blood cells and determine whether they're higher or lower or within normal range. And that's called a differential white blood cell count. Okay, so let's look at some of the characteristics. In general, there are two types of white blood cells. We have what are called granular leukocytes or granulocytes, and we have agranular leukocytes or agranulocytes. Now, the granular leukocytes are called that because in their cytoplasm, they have little granules which will secrete certain substances. And depending upon the cell, the types of granules they have, they can fight different types of infection. So, granular leukocytes, they help to detoxify foreign substances. They also release heparin, which is an anticoagulant, prevents clotting. The three types of granular leukocytes are neutrophils, eosinophils, and basophils. So what I want to do is look at each one, and we're going to refer to your handout here. So let's start with the neutrophil. Okay, the neutrophil is the last one listed under granulocytes. The neutrophil is called the neutrophil because its granules are neutral in terms of the type of dye that it accepts. So the granules generally look kind of a purplish color, gives the cytoplasm kind of a purple color. Under our magnification, we probably won't see that so well, but the one thing you will see is the nucleus. The nucleus can be in different shapes or morphologies. It can look different, but that's like the most distinctive thing. It has multiple lobes to it, which are kind of attached by these little tracts of tissue. So the neutrophil, again, it is kind of a lilac-colored cell, and it has this nucleus that has many different appearances. We call that poly for many, morpho for appearances, nuclear for nucleus. So our nucleus is polymorphonuclear. So one thing. Yeah, they're not multinucleate. They have one nucleus, but it has multiple lobes.
Oh, I get it.
But I don't know if you remember me mentioning when we looked at the blood smear. I said that this tissue is stained with something called a Wright stain, W-R-I-G-H-T. Remember, there is an acidic dye and a basic dye that are going to be attracted to the different components of the cells. That's why, you know, the white blood cells have different colors, the red blood cells are red. So, like I said, the neutrophil, the granules are neutral. They accept both the red and the blue dye, so that it kind of looks purplish in color. Now, neutrophils are the most abundant white blood cell type that you're going to find, by far. Neutrophils contain granules that favor both acidic and basic dyes. They stay pink in color. They're polymorphonuclear. They're the first cells on the front line in an acute infection, and that infection is usually bacterial. It's usually a bacterial infection. Most abundant cell type in the blood. Then we have the eosinophil. Eosinophils in the cytoplasm, the granules, they stain red. because they have an attraction for the acidic stained eosin. They're called eosinophils. The nucleus in all these cells always are going to be blue. The nucleus is always blue, but the granules will be different. So eosinophils, if you see a big red cell in there with a blue nucleus, it's an eosinophil. Generally speaking, they they secrete substances that are important in fighting parasitic worm infections, but you also see them in higher numbers when a person has an autoimmune infection. So for example, or an autoimmune condition. So if a person has, for example, lupus, they may have a higher than normal number of eosinophils. Basophils, the third type, is going to have granules that are attracted to the basic dyes, which make basically not just the nucleus dark blue, but also the entire cell dark blue. Basophils are the least abundant white blood cell. The granules that they have secrete histamine, which is important for kind of counteracting allergies and things like that. But unfortunately, histamine is what causes your watery eyes, your blood vessels to dilate, and all of that, which is why you take an antihistamine to prevent the effects of the basophils. All right, then we have the agranular, or agranulocytes. Here we have the two. There are lymphocytes and monocytes. So first of all, the monocytes are probably the largest white blood cell. They're very big. Monocytes they have no granules. They're agranular. Their cytoplasm looks very kind of grayish blue, and they usually have a very large dark purple nucleus. They say it looks like a horseshoe, but many times I don't see it looking like a horseshoe, because a lot of times when you see a monocyte, it almost looks like it's going somewhere. Like you guys remember from the video this morning how the white blood cell was slinking its way through the capillary, and then it left the capillary through a process called diapedesis. That's where it flattens itself out and moves into the tissue space. Monocytes are usually on the move like that, so they might not even look circular, they might look more oval. And the monocyte becomes a macrophage when it gets into the tissue spaces. So what we looked at today when we were looking at phagocytosis, remember? We were looking at bulk transport, and I said this is a macrophage that's ingesting a bacterial cell. It's actually a monocyte that's moved into the tissue spaces. They usually go after bacterial infections if they're out in the tissues. You'll see a larger number of monocytes. All right, and then we have the lymphocytes, which are really tiny. They are pretty much all dark purple nucleus with a little bit of cytoplasm around them. Again, they're agranular, so you don't see any granules. Now, these can differentiate into two types of lymphocytes. They can be T cells or B cells. B cells will actually make antibodies that attack bacteria and toxins, but T cells can differentiate into different types of T cells, including the natural killer T cells. They can cause cells to trigger apoptosis. That's important with cancers and things like that. Or cytotoxic T cells that destroy cells that are virally infected, or tumor cells. So lymphocytes you're going to find in the lymph nodes a lot of times, in the lymphatics. And so lymphocytes are very important little cells. But here's an interesting thing. You know mononucleosis, right? You've heard of that. Now, the name may make it sound like, you're going to have a lot of monocytes, but in fact, it's not. Remember that monocytes go after bacteria, lymphocytes go after viruses, and mono is caused by the Epstein-Barr virus. But what happens is when they're active in infections, they can appear larger like monocytes, and so in a way this was sort of misnamed. So with mono, you're going to have an increase in number of lymphocytes, not monocytes like you would think. Okay, so any questions so far about that? So I mean, one thing, if you take a look at page 3, the specific white blood cell number indications I have listed here for you, and it goes along with, you know, the general descriptions that we just talked about. So it says here, you know, with neutrophils, the most abundant white blood cells, says the percentage is often increased, which specifically is called neutrophilia, during an acute bacterial infection. Interestingly, last trimester of pregnancy, some fungal and viral infections, necrosis, like if a person's had a heart attack and some of the cells have died, or if you've had burn and there's a burn and there's a lot of tissue to clean up, the neutrophils will go after that. A low number, neutropenia, increases the risk of bacterial infections, which can be from bone cancer or an overwhelming infection in the blood, septic infection. Eosinophils, we mentioned, a high number is known as eosinophilia, indicates parasitic infections, inflammatory diseases, as we said, like autoimmune, like celiac disease and eczema or asthma. Low numbers, eosinopenia, generally because you're gonna see, like with the eosinophils and the basophils, they're such a small number that small numbers of them aren't necessarily medically significant. If we look at basophils, if you have higher numbers, it's called basophilia. It's found with inflammation, allergies, and damage of the tissues of the body. Lower numbers is not significant because basophils usually find less than 1% of them in the blood. And then we can see monocytes and lymphocytes. Monocytes, I mentioned, they mature into macrophages, which are important for destroying, you know, bacteria and things like that. Monocytosis is when you have an elevated number. You find it with TB, tuberculosis, bacterial infections like bacterial endocarditis in the heart, or autoimmune diseases like lupus. Monocytopenia is where you have very low numbers, can be seen with bone marrow damage or leukemia. And lastly, lymphocytes. High numbers, known as lymphocytosis, is found with viral infections like Epstein-Barr, which causes mono, or the chickenpox virus. Individuals with hepatitis or herpes also have an increased count. Low numbers, known as lymphopenia, can be seen with autoimmune diseases like lupus or RA. So, you know, if a person has RA, for example, or I should say lupus, they'll have a larger number usually of monocytes and lymphocytes, but the other numbers might be okay, provided they don't have a, you know, a problem. Back in the day, I used to be able to, before I was told I'm not allowed to use real blood, I had the students actually do blood smears when we did right stain. So we had them prick their fingers and they would smear, and then we do a right stain and actually prepare the blood smear. And I remember one time this girl who was in the class, she had a high number of lymphocytes, and she's like, I don't know if this is right. I said, well, I think it looks right. And the next week she was diagnosed with mono. It was so interesting, you know, like she got herself. But we've had people have, one time we had somebody who had a heart defect, an arrhythmia, and we picked it up, which isn't common because we don't use a 12 lead. And then one time I had a girl who had a urinary tract infection, and we caught that. So we found out, so in our small classroom lab, we used to find all kinds of stuff.
Were those diagnosed like in class, in front of the whole class? Like if I had a urinary tract infection, I wouldn't want everyone to know, maybe. Maybe not. I mean, I personally don't care. But that girl might know it, yeah.
She didn't like yell it out. She told me privately.
Oh, that's what I'm asking. Yeah, if it was like, hey, look, Keisha, this is my cat. You're like, hey, turns out
Yeah, no,
you
I
have
would.
this.
No, I would never say. Yeah, no, I wouldn't know. But then she gave me more details I really didn't want to know. So sometimes it's like, you know, people tell you stuff and, you know, they say it and I'm like, you know, well, it could be this, you know, quietly. And then they get loud and they talk about other things. It's like, okay, well, all right, you know.
He told me he used protection.
Because apparently in this class, learning aseptic sterile technique, why is my tongue like all twisted today? isn't part of the class. It's part of micro, but it's not part of a class like this. This is actually a clean lab, so we can't.
Can't be slicing and dicing in here.
Can't we just go to the other lab?
No, the idea is it's not part of the curriculum. It's not actually part of like the objectives of the class that we learn that. And no, I agree. I think it would be much more fun, and I loved doing that. I thought that was so much fun. I really did. Anyway, okay, so lastly for this handout, I just wanted you to see, this is sort of a preview of what we're gonna do when we come in on Thursday after the quiz. This activity 1 is we already did activity 2. So activity 1 is, again, performing a differential white blood cell count. So for this, you probably want to use the poster, because the poster, it's higher magnification than what we can do with the light microscope, but you can use your oil immersion lens, and that will give you greater detail when you do this, when you're trying to identify them. And this shows all you do with a differential white blood cell count. This is where you're going to use your cell counters in a purposeful way, I guess you could say. If you see, for example, so you're going to count 100 white blood cells. And if you see, when you're counting, you can see a basophil, you click 1. If you see a neutrophil, click 2. Another neutrophil, click 1. So you keep clicking on the cells that you see. So you'll start at the top of the slide, kind of work your way down, move over, go up the slide. So you kind of snake your way through the slide. And then when you get to 100, you're finished. And what you'll do, for example, if you saw 50 neutrophils, that means you had 50%. If you had 1 basophil, you had 1%. And you can write down your percentage and compare the normal range. So it's very easy to do. The trick is to be able to identify which cells you're looking at. And it's a good activity for us to get used to looking at the way the cells should appear. So generally speaking, if you want to remember the order of the most abundant white blood cell to the least abundant, you can remember the phrase, never let monkeys eat bananas. Neutrophils for never, lymphocytes for let, monocytes for monkeys, eosinophils for eat, and basophils for bananas. Bananas for basophils, I guess. It's very easy to do it that way. So that's what we have going on for us coming up. So let me just go through a couple more here, because I do want to give you guys a chance to do the white blood cell stuff. But I also did want to talk about a few other things, which we're going to have to get to definitely in the next class. Okay, so we talked about the granular and agranular leukocytes. Okay, and platelets. Platelets are important. They're not really cells. We mentioned this before. They're not really cells, but they are fragments of a large cell called a megakaryocyte, not shown here. But it is a very huge, huge cell whose cytoplasm is sort of partitioned off. And what happens when we eat platelets, it just sort of peels off parts of itself to make these platelets, which is very interesting in itself. But the megakaryocytes come from the red bone marrow, just like all the other cells do, the red blood cells, the white blood cells. And the platelets constitute most of the mass of blood clots. Platelets release serotonin, along with a couple of other chemicals we're going to be talking about. But the serotonin specifically helps to constrict and reduce blood flow to the clot area, because if we cut ourselves, we don't want to keep hemorrhaging. So we're not going to like dilate our blood vessels and get more and more blood to the area so we can lose it. We're going to constrict it, and serotonin helps with this. Also, they secrete growth factors that help. When a blood vessel wall is healthy, it helps to maintain its integrity. But platelets only live about 5 to 9 days, because we're always making more. And of course, if we had a high platelet count, that could increase the risk of clotting. So you have to watch for that. Hematopoiesis is the process by which we produce blood cells. Hema means blood, and poiesis means production of. So hematopoiesis is the production of blood cells from stem cells in the red bone marrow, also known as myeloid tissue. You may hear the word myeloid. You talk about, like, for example, a myeloma. That is a cancer which is in the red bone marrow. So anytime you hear like myo as a prefix, that indicates that we're talking about myeloid tissue, which is the red bone marrow tissue. And also lymph tissue. Lymph tissue also helps to make some of our blood cells. Our red bone marrow produces about 500 billion blood cells per day. And in the fetus, because a fetus's skeleton is made up of what kind of tissue? Remember? Hyaline cartilage. It doesn't have a marrow cavity. It doesn't have any cavity. So in the fetal body, it's actually producing its blood cells in the liver instead of in the bones until, of course, it gets older and then it has ossification that occurs. Okay, so hematopoiesis, we have two specific types of blood cell production. We have erythropoiesis and leukopoiesis. Of course, erythropoiesis is specifically the production of the red blood cells, and leukopoiesis production of the white blood cells. Erythropoiesis is stimulated by EPO, like we talked about in class on Tuesday. EPO is released from the kidney, goes to the red bone marrow, and causes the myeloid tissue to generate more red blood cells. Why do we release EPO? What's the reason for this? Because we have what? Why is EPO released in the first place? What would you need more red blood cells for?
Fight off infection.
Nope, red blood cells don't have anything to do with infection.
To heal. I don't know.
Filtration.
Not filtration, no. But red blood cells, they do what? What's their main job? Carry oxygen.
Oh,
We just don't have the ability to carry around enough oxygen, right? We don't have enough. Maybe we're anemic, maybe we're at a high altitude, something like that. So that's going to trigger the kidneys to release the EPO. Whereas leukopoiesis is going to happen because we need more white blood cells to do what, Anthony?
Fight off infection.
Fight off infection, because that's what white blood cells do. They're involved in immunity. This is stimulated by a variety of cytokines. It's basically a cascade of events. We're not going to get into the particulars here because this isn't an immunology class, but basically these cytokines are going to trigger what kind of white blood cell we need to make more of in order to be able to fight the infection that we have, whichever type it is. And so that's, and actually the chemical, and it is listed on the first paragraph of this lab printout. The chemical which is responsible for triggering leukopoiesis is known as CSF, colony stimulating factor. And if you look at the second to last sentence in the introductory paragraph, it lists CSF there. You might want to highlight that, because perhaps I'll ask a question about that. Which chemical stimulates leukopoiesis? And that would be colony stimulating factor. Let me see here. Okay, yeah. So I think what we'll do, we'll finish this slide and then we'll call it quits for today. Like I said, we'll come in, take our quiz, do the differential white blood cell counts, and then we'll talk about clotting. Call it a day Thursday. Tuesday next week, we'll come in, take our other quiz on red and white blood cell labs, and we will talk about blood typing, and that will be probably the most fun lab. And then after that, we don't have class next Thursday because it's 4th of July. Tuesday after that, we have our lab midterm. And I will be giving you guys the study guide for the lab midterm, probably end of this week or the very beginning of next. All right, so last slide. Erythropoiesis, this is just showing us red blood cell production here and the stages that we go through. So we make about 2.5 million red blood cells every second. That's pretty astounding. But they live 120 days. We always need to have more to take over from the old ones that died. We did talk about in the last class how red blood cells die, what happens to the components of hemoglobin and those types of things. The old red blood cells are removed from the blood by phagocytic cells in the liver, spleen, and bone marrow. And we know that globin is hydrolyzed. The heme is usually going to be degraded into strocobilin, which is the brown pigment. Remember in feces, we talked about that. And the iron is going to be recycled back. The body doesn't like to get rid of iron if it can. Although, if you drink a lot of alcohol, you do tend to flush out iron as well as other nutrients in your body. So that's just sort of a side effect of, like, you know, if somebody drinks a lot of alcohol, it can lead to iron deficiency, a little bit of iron deficiency among other deficiencies. So anyway, we start out with the hemocytoblast, which is a stem cell in the myeloid tissue, and we have EPO stimulating something called a proerythroblast. It's like a baby red blood cell. It is going to be stimulated by EPO to become an erythroblast. So in other words, it basically triggers this cell to develop along the lines of a red blood cell. That's what EPO does. Because this cell could technically be any type of white blood cell too, because these are stem cells in the myeloid tissue. We go from being an erythroblast to a normoblast to a reticulocyte, and finally an erythrocyte, which is finally released into the blood. Now, what do you notice about this, which we know is strange for a mature red blood cell? What do we have here that we know we don't have in a red blood cell? In this part here, what do you see that we know isn't in a red blood cell? There's two circles there. One circle shouldn't belong. Which one? Huh?
The outside, the surrounding.
The nucleus.
The nucleus part, right? Red blood cells don't have a nucleus, they can't reproduce. But we start out with one, but then it gets smaller and smaller. Why? Well, basically, once we start making more and more hemoglobin, because we need the DNA initially to make the hemoglobin, once we start filling the cell with more and more hemoglobin, the nucleus gets smaller and smaller until once we get into the normoblast, before we become a reticulocyte, the normoblast expels the nucleus, and then we have a nucleus-less cell that's produced. All right, any questions?
So we can know that.
No, it's okay. You don't have to know. What I would just say for this, in terms of knowing this, I would know that EPO stimulates it. Initially, we have a nucleus. We end up not having one. That's the key thing in that particular slide. All right. Any other questions? Okay.